topic is about synaptic density and clinical use of PET in Rett syndrome. Before you ask what Rett syndrome is, let me tell you about my research opportunity and interest. So last summer, I was lucky to be elected into the research fellowship program coordinated by Lawrence University at Weizmann Center. Here's a picture of Lake Manhattan at Madison. It's a very nice and beautiful city, especially during the summer and sunset. These are signature chairs at the terrace. This me at the lab, and this Weizmann Center, where I work at the lab on the 6th floor. So Weizmann Center is dedicated to advancing knowledge about human developments, developmental disabilities, neurodegenerative disease through research, training for faculty, providing service for those who need and outreach to the community. So my first term of freshman year, I took psychology class, and I was very interested in human's brain and how different parts of human brain work and to perform the function. Further study, I'm more interested in neurons and how they communicate with each other by synapses and neurotransmitters, and how genetic change can change the morphology and function of the brain. So Rett syndrome is a rare disease, and it's important to study rare disease because people who have rare disease also need care and attention. Rare disease is often caused by a single mutation of the gene, so it makes it easy to manipulate experimental model for research, and also scientists can learn other common related disease and the genetic mechanism by studying the role of a single gene. So what Rett syndrome is a genetic and neurodevelopmental disorder. It's often misdiagnosed as autism or cerebral palsy. This is a picture example of girls with Rett syndrome. It affects all races and ethnic groups, often in girls, 1 in 10,000 to 15,000, 0.01%. Early onset is a normal period of growth and then show regression of development or abnormal sign and behavior. So some of the clinical symptoms of Rett syndrome, there are 3 criteria to diagnose someone with Rett syndrome. The first one is essential criteria, must present for diagnosis. It includes progressive loss of motor skills, or like body movement and speech, intellectual and developmental disability, gait ataxia, which is uncoordinated body movement or loss of body control, stereotypic hand movement, which is repetitive and purposeless. It includes hand wringing, grabbing, tapping, squeezing, or like rubbing. The next criteria is a supportive criteria. It's not necessary, but often present. It includes scoliosis. This is a picture of girls with severe scoliosis with a very great angle of curve that could affect her lung. They can also have respiratory distress, breathing irregularities, hyperventilation, or like air swallowing, abnormal muscle tone, delayed growth, cold and small hands and feet due to poor circulation, epileptic seizures, and reduced response to pain. The last criteria is exclusion criteria. So it rules out Rett syndrome possibility. It includes other inherited degenerative disorder, severe infection or head trauma, brain damage, or abnormal acquired after birth, or abnormal development for the first 6 months of life. So what the brain of a patient with Rett syndrome looks like, it was found that compared to the control normal brain in the people with Rett syndrome has decreased brain volume. Here's a MRI scan with different layer of brain cut. So specifically, there's a decrease in cerebral volume, which is the outer gray matter outside the core brain. Selective decrease in parietal lobe is shown in red circle here. Parietal lobe is important in processing speech, math, and sensory of the body, such as like location, parts of the body. It also shows mild reduction in white matter and reduction in anterior frontal lobe. So it correlates with clinical severity. People with more severe Rett syndrome will show more reduction of brain volume in this area, shown yellow. And this area is important for thinking, planning, and reasoning. Here's a closer picture of patient with Rett syndrome compared with the control. So this is a negative image. So that means like the white color here show gray matter, and the black color here show white matter. So compared to the control, Rett syndrome brain show the decrease in both gray and white matter. So what it means by reducing brain volume at cellular level, it can be that the total amount of neuron is decreased, or that each neuron cell is smaller compared to the normal neuron, or there's less synaptic connection, which my project specifically look at. So synapses is important to tell the brain activity, and also in other neurological diseases such as Alzheimer's or depression, they found there's a regression of brain activity and a decrease in the synapses. So to look at the synapse, we'll look at the neuron, which is a single unit cell in the brain. Just like any other cell, it has nucleus and the cell body, also soma. In addition, it has a dendrites. Axon appears white color because it's grabbed by myelin sheath. Axon terminal, including synapses, connect the axon terminal with the next, the dendrite, the next neuron. So in short, it's a single unit major cell type in the brain communicate with one another via transmitting chemical and electrical signal. So synapses is the connection between neurons. Here's a sending neuron sending action potential to the next neuron by synapses. Here's an animation of the transmitting signal. Closer look at the synapses. We see that here is axon terminal, which is a presynaptic cell. Here is a dendrite postsynaptic cell. So the synaptic vesicle packed with neurotransmitter is transported near the membrane of the presynaptic cell, and then fused with the membrane and release neurotransmitter into the synaptic cleft. Then the neurotransmitter binds to the receptors to induce further downstream signaling for action potential. So synapses can form where there is a dendritic spine. So what is a dendritic spine? It's on the dendrites of the neuron. It's a membrane protrusion from the dendrite that form the mushroom-like structure. It can form the synapses with the axon terminal of another neurons. So in Rett syndrome, they found that there is lack of dendrites and dendritic spines. So here is a normal maturation of the neuron with the fully grown dendrites and dendritic spines. But in Rett syndrome, there is an incomplete maturation. We can see that there is lack of dendrites and dendritic spines. So it refers that this could show the possibility of reducing synapses. due to the lack of genetic spikes. So what causes the abnormal growth in neurons? Rett syndrome is majorly caused by a mutation of one gene. So it's a mutation in X-linked gene called metal CpG binding protein 2 or MECP2 gene. This gene is on the X-linked X chromosome. It's a sex chromosome. It regulates gene play a role in normal brain function, particularly maintenance of synapses. So here we have the healthy cell with normal MECP2 on, producing normal MECP2 protein and to do its function. But in the mutant cell, normal MECP2 is off. There is no normal function MECP2 protein, and the mutated MECP2 is on, producing MECP2 protein with disrupted function. So what I mean by on and off here is a gene expression process. So here we have the normal MECP2 gene on. We have the DNA located on the chromosome, which is double-stranded DNA. It transcribed into the single-stranded RNA and then translated into protein. And normal function of MECP2 protein will perform its function of normal neuronal growth or dendrites. Next, in the mutant cell, normal MECP2 is off. We still have the DNA double-stranded, but there's no transcription or translation, and there's no MECP2 to perform its function. Also in the mutant cell, we have the mutated MECP2 is on, and the DNA is transcribed to RNA and then to translate to protein. And then the mutated MECP2 protein with disrupted function will express the Rett syndrome phenotype. And MECP2 is a transcription repressor, and how it regulates other genes to express the Rett syndrome phenotype is through the transcription process. We'll look at the transcription complex. Here's the double-stranded DNA with the promoter region shown in dark green color. Transcription factor will bind to this promoter region and recruit RNA polymerase to form the transcription complex, open the double DNA template, and then transcribe the RNA transcript based on this one template here. Here's the animation of transcription complex. The template double-stranded DNA coming in, and then the single-stranded RNA product is excreted. Here's still the template going out. So MECP2 is the metal CpG binding protein. It binds to the metal CpG island. Metal CpG is on the DNA sequence. It's a dinucleotide of cytosine followed by guanine, connected by the phosphate group. The enzyme will add the metal group to the 5' cytosine, and methylation of DNA will make the DNA sequence inaccessible to RNA polymerase and prevent transcription. So in mammals, 70 to 80% of CpG cytosines are methylated, and in humans, 70% promoters located near the transcription start site of the gene contain a CpG island. So we'll look at the inappropriate gene sequence here. There's a gene sequence of inappropriate gene with the transcription start site shown in the arrow. Near the promoter, there's a methylated CpG island, and the white is an unmethylated CpG. So MECP2 will bind to the methylated CpG and recruit other transcription repressors such as HDACs and prevent transcription of the inappropriate gene in the normal cell type. However, in the Rett syndrome cell type, the absence of the MECP2 or mutated MECP2 cannot bind to the methylated CpG, leaving space for transcription factor and polymerase to bind and to transcribe this inappropriate gene. And Rett syndrome phenotype expressed. So we know that the major cause of Rett syndrome is this single gene. Now we will manipulate this gene to create the gene-modified mice model to characterize synaptic density, including dendritic spikes. Another goal of my project is compare the histological result of both with the result using PET scan to validate potential use of PET at clinical and for research, so that we can reuse mice again and again. Next is a method. So we use mice model because it has 85% of similar protein coding sequence with humans. It's cheap, it's easy to handle, breed and maintain. It just needs small space to live. It has short lifespan, 2 years age, produce 5 to 7 pups per birth, and less ethical concern than other animal models such as non-human primates, dogs or cats. From these mice, we will remove the MECP2 gene by technique knock out MECP2 gene. To do that, we will first isolate mice embryonic stem cell from the blastocyst, and then we introduce the artificial DNA sequence, which is similar to the MECP2 gene, but the upstream and downstream sequence are shown in yellow block. This gene also contains a marker gene that confers a resistant or produce observable change of fluorescent light or color. The gene is introduced by electroporation to increase the permeability of the cell. So this artificial DNA sequence will line up with the MECP2 gene and integrate into the gene sequence in the cell, or replace the MECP2 gene by homologous recombination. So the end result is that in the cell there will be artificial DNA integrated and the MECP2 gene is removed out. Because this process is happening randomly, so we need to select the cell that successfully modified gene by resistant marker. When we have a group of cells that successfully gene engineered, we introduce back to the blastocyst. We introduce back into the blastocyst and then transfer into the uterus of host female. Her offspring will have heterozygous MECP2 gene removed. After we knock out the MECP2 gene mice, we will grow, collect the offspring, and genotyping to determine which gene mutation it has. Then we use them for PET scan for data collection. With the same group of mice, we will euthanize and mix the sliced brain and look at the synapses of hippocampus. So hippocampus in human shown in red circle here. It looks like a seahorse structure. It's a part of the limbic system, which involves in motivation, emotion, learning and behavior, and also memory. Hippocampus specifically important information of spatial memory and navigation, learning and behavior. Mice hippocampus is evolutionary conserved in morphology and also in function with humans hippocampus. hippocampus in mice also extensively studied for neuroscience research because this brain region is really activated when the mice is put into the behavioral assay so that scientists can learn activated brain region and also compare with the behavior data. And then we'll make the brain cut, very thin brain cut section. Here is a hippocampus at two sides of the brain. In this picture, you can see locate the hippocampus by the very thick white line here at two sides. Then we incubate this brain section with antibodies, which is produced. It's a Y-shaped protein produced by plasma cell. It's neutralized pathogen by binding to specific epitope of the antigen. Here's a Y-shaped protein with the heavy chain shown in blue and red chain, light chain shown in red. It has the antigen binding site which is specific to specific antigen. It binds only to A but not B or C. It's the acquired and specific immune response. What I mean is it has the immunological memory leading to the enhanced response upon second exposure to the pathogen. Because of its specificity binding to the antigen, scientists employ this to design the antibody to bind to the protein of interest in order to visualize it. So how we identify or visualize synapses? Because synapses is actually a gap or is a cleft. Here, if we look back here, the synaptic vesicle needs some protein complex in order to bring to the membrane and to fuse with the membrane of the presynaptic cell. Here's a demonstration. So we have the synaptic vesicle with the protein complex that help it fuse with the membrane and release neurotransmitter. So we will use an antibody to tag to the what kind of protein in this complex. So here we have this presynaptic protein. We will incubate it with the primary antibody with the specificity, and then the second antibody with the fluorophore molecule binds to the primary antibody, help visualize by fluorescent color. So fluorophore molecule, here's an example of fluorophore molecule. It's a fluorescent chemical compound that can re-emit light upon light excitation. It contains aromatic groups, benzene ring here, or planar or cyclic molecules with several pi bonds. Here's the pi bonds. Here's the energy chart of the fluorophore molecule. So at the ground state, fluorophore molecule will absorb light at specific wavelength to have higher energy to become the excited state. However, it will lose some of the energy due to the vibrant structure of the benzene ring and resonance structure. So it will lose some of the energy at the process too, and it will emit the fluorescent light when the molecule come back to the energy ground state. And it emits the fluorescent light at longer wavelength. Thus, it will produce different color compared to the initial color it absorbed. So here we have the excitation and emission spectra of nucleotide called data. Here's a fraction of light absorbed or excitation at the wavelength purple color, about 400. When it emits the fluorescent light, it will emit at the longer wavelength, show in blue color here is about 500. So one application of using fluorophore molecule is staining HeLa cell. An example, DAPI show in blue stain the nucleus, and the red and green color is staining protein, structural protein. So we have the brain cut section. We do the fluorescent immunohistochemical assay by incubating with 2 antibodies, and then look under the confocal microscope. The expected image show here with the synapses. The red color stain the presynaptic protein, and the green color show stain the postsynaptic protein. And the distance between them is about 20 nanometer. After putting under the confocal microscope, we will capture a lot of image in order to build the 3D image by the software Imaris. So here is a 3D picture of the brain cross-section. With the blue show nucleus, DAPI staining. The red and green is puncta or the synapses stain. Here is a picture of only red and green channel that we hope to look at. This is a picture of Imaris 3D selection. So we will run the software, and the co-localization of red and green color will be selected by the software. And this is qualified as one synapse if the red and green color is close to one another. So should I take over? Yeah, I will turn off the light for this slide. So here's a picture. This is a video. Just see how is it. So this is a 3D picture of the synapses. And this how dark I work at the computer room in order to see clearly. Then we have collect the results of number of synapses, and from the histological analysis, we found that there's a synaptic density is decreased in Rett syndrome. So compared to the wild type, which has 452 red and green co-localization in the y-axis. Rett syndrome has 237 red and green co-localization, about half of the amount. However, we didn't achieve the significant p-value because the data is varied. We then compare this histological data with the PET scan data. So PET is a positron emission tomography. We have the cyclotron here to accelerate particles. Positron emitting radionuclides are made by attaching radioactive atom with the molecule that brain used for metabolism and activities such as glucose. Then it will be created into the radio chaser and injected into mice. So in the mice brain, where there is more activity, more radionuclides will get there to the brain, most activated brain area. So the radionuclides will emit the positron and will collide with the electron in the mice brain. This collision will release a photon at the same energy level by process annihilation. This energy level will be detected by PET machine. And after a lot of math, complex math equation, the image will be produced and constructed. Here's the image of PET scan with a high brain activity shown in red color. Gradually, green, blue, and dark blue is the low brain activity. So PET is good temporal because it shows brain dynamic or functioning. It can have neural or biochemical radioactive chaser. It's cheap and high specificity because researcher can design the radioactive atom that specifically bind to the molecule that they want to look at. So we have the PET scan data. X-axis is a time in minute, Y is the concentration of radioactivity. Compared to the wild type, we show in red. Rett Syndrome sample shown in blue show the decreased radioactivity. For the comparison of the two results, we have the implication that first, the synaptic density is decreased in Rett syndrome, impeding learning and brain activity. And second, PET is a promising tool to study brain activity at cellular level and at clinical. For future direction, we hope to collect more mice brains sample for more data collection, and also more male mice to put into the data because male mice is very sick and die at young age. We can also use different presynaptic or postsynaptic protein for the future study. Acknowledgement. I would like to thank Lawrence University for research fellowship opportunity, Professor Knudsen, Professor Hill and Ellen West for coordinating the program, Weizmann Center and Professor Chang. Postdoctor Dong, Research Associate Jan Hong and other lab colleagues, Advisor Dr. Dev, and Dr. Ramos for some of the neuroscience concept. I thank Dr. Dev for giving me very detailed advice on organizing the slide and hear me practice again and again. I would like also thank my friends and my family, my pets and Google for make this slide happen. So, thank you for being a great audience. And any questions?
Is there any other hypothesis for the cause of Rett syndrome besides next feature?
Yes, it's a good question. Well, so if we look at this one, if there's a mutation of that causes the CPG island disappear from the gene sequence, or something wrong with the enzyme that cannot add the methyl group to the CpG island, so the MAC2 protein cannot bind to the methylated CpG. This also could be the cause of the Rett syndrome. Because the goal here is to have the MAC2 bind to the methylated CpG and prevent the transcription. So any happen to the CpG island or the MAC2 can produce the same consequence. Any other question?
So what happens if you use like an antidepressant that stimulates neurite growth in this Rett syndrome model? Will there be more synapses formed? So could that be, could you use an antidepressant or something like ketamine to stimulate dendritic spine growth and then ameliorate the effects of Rett syndrome?
Yeah, so it's actually like the pharmacological treatment for Rett syndrome is having drugs that or like some kind of growth factor to promote the growth of the neurons. It helps the morphology of the neuron, but it's probably not treat the cause of the syndrome because it's like genetic mutation. So yeah, there is also like genetic therapy, but it's expensive and not common.
Any questions? Great, let's thank our speaker again.